Oh, wait. Just had to click away the recording notification. Okay, so good morning or good afternoon or good evening, depending on where you are right now. Well, I can say I'm currently in Germany and it's already pitch black outside. So thank you very much for inviting me today. It's a pleasure for me to be here. Our work mainly focuses on the discovery and optimization of specialized metabolite pathways, as Lars already mentioned, in a wide range of food crops, bioenergy crops, and medicinal plants. And today I'd like to talk about one of our projects where we focus on how to discover the genetic basis of specialized metabolism in plant stress. So we all know that we are using more energy than what's sustainable for our planet. And one approach to tackle this imbalance is the production of renewable energies, including biofuels. Here in Europe, the main bioenergy crop is rapeseed for biodiesel production. But switchgrass and other crops are on the rise. In the US, the major crop for biofuel production is corn. But the criticism here is that if we use corn for biofuel production, we're taking it away from the food production chain. And that is why it became necessary to start researching alternative bioenergy crops like switchgrass. And some of the reasons why switchgrass is so successful as bioenergy grass are its high net efficiency, that it grows on marginal lands and doesn't compete with food crops, that it can be harvested repeatedly, and it has a very high biotic and abiotic stress tolerance. But there are also some challenges to it. For example, that it has an allotetraploid genome with a high variation across the different varieties and even between individuals. It's an obligate outcrossing species, and like those challenges combined makes it very hard to apply common gene editing techniques like CRISPR, for example. And there are two different ways that you can make biofuel from switchgrass. One is through the production of bioethanol from lignocellulose, and the other way involves a group of special metabolites that probably all of you are more or less familiar with, the terpenes or so-called terpenoids. Terpenes or terpenoids are a chemical group of diverse small molecules. The building block for the terpenes, for all terpenes, are small C5 isoprene units. And these building blocks can then be combined into a wide range of C10, C20, C30 molecules, which are then called monoterpenes, diterpenes, triterpenes, etc. And some of those molecules are acting as precursors for plant growth hormones like the gibberellins, whereas others function as specialized metabolites, which means that they're not essential for the plants, but mediate interactions with the environment, for example, in friendly ways by attracting pollinators, and those are mainly volatile mono and sesquiterpenoids. Sesquiterpenoids are the C15 compounds, or by facilitating a healthy microbiome, which is essential for the plant fitness. For example, this group of diterpenoids found in corn, which are called the dolabrolexins, but also in not so friendly ways. For example, by deterring herbivores. And here you can see an example from the Bohlmann lab, the diterpene resin acids, which consists of diterpenes. And it's estimated that plants can produce more than 80,000 of those different terpenoids, and that's only what we assume so far. But they're not only useful for plants, but are heavily utilized by us humans too. So here's some examples how we are using terpenoids in our everyday lives. If your toothpaste tastes like mint this morning, then you're probably having menthol in your toothpaste. And the more volatile monoterpenoids like limonene are often used as fragrances in your perfume and bath and cleaning products. But also pharmaceuticals, for example, artemisinin, which is a popular drug against malaria. Or here you can see Taxol, which is used as a chemotherapy drug and is originally found in the Pacific jute tree, or even as biofuels, here as bisabolene or caryophyllene, which can be used as jet fuels. And even though there's a lot of interest in the discovery and engineering of the pathways involved in terpene metabolism, progress has been somewhat slow due to the huge diversity of those molecules and the often very species-specific occurrence that requires us to work with the natural producer plants and not just rely on established model plants. So fortunately, new developments in synthetic biology, among others, have made great progress toward new approaches for the functional analysis of enzymes involved in terpene or general plant specialized metabolism. So here I would like to present you the workflow that we are using to characterize genes, for example, in switchgrass. So the common starting point are public genes and gene and protein databases, and then you blast those databases against signature protein motifs and domains to collect this initial pool of gene candidates. These gene candidates then must be filtered for pseudogenes and other misassembled transcripts, for example. In addition, you can look for gene clusters or do phylogenetic analysis, which can also help with the candidate selection to narrow this pool down. And then luckily, DNA synthesis has become increasingly cost effective and now gives us access to transcripts that are naturally difficult to amplify. So we get our selection of genes synthesized into the desired vectors. And now comes the verification of gene and enzyme functions through either genetic or biochemical approaches or a combination of the two. And once we have found the gene or enzyme function, we can then feed this information back into the databases. And with the help of this pipeline, we've been able to characterize what we know so far about the diterpenoid network of multiple food and bioenergy crops, including switchgrass. Diterpenoids are the C20 molecules, and switchgrass has the biggest so far known family of diterpene synthases in any monocot plant. Also, after narrowing down the huge candidate pool to about 30 genes, we got them all synthesized and could then use biochemical approaches to characterize them. Here you see the typical pathway from the precursor molecule geranyl pyrophosphate, which undergoes cyclization by a class 2 diterpene synthase. And then consecutive, the class 1 diterpene synthase modifies this further. And then in many cases, cytochrome P450 enzymes further decorate those molecules. We are currently characterizing some of the over 100 P450 candidates genes we have identified. So as you can imagine, this will really add up to the diterpenoid network mapping. Here you can see an example of some recently published functions where the P450 enzymes directly act on the class 2 products, which is somewhat uncommon. But that also can enhance the amount of possibilities you have to create new molecules. But yeah, so we have to assume that those are not necessarily the end products because we know that there's probably more. But now that we discovered all those enzymes and know what products they make, the real question is what for is switchgrass making all those diterpenoids? And from other crops like corn and rice, we know that diterpenes act as defenses against pests and pathogens. And we are collaborating with the Northern Lab at Lawrence Berkeley National Lab in California to test the influence of selected diterpenes on switchgrass microbiome. So please check out ecofab.org. It's a very cool project. From other crops, we also know that diterpenes can mediate abiotic stressors like drought stress. And drought stress is one of the many problems that is increasing with climate change and is a big threat for many major crops. Drought stress is actually the main cause of yield loss currently in the world. And that is why there are many efforts currently trying to make our crops more drought-proof. And one way to do that is to compare closely related ecotypes of the same species that have adapted to different environments, and then identify stress tolerance related candidate genes by comparing the expression profiles of the different ecotypes. So switchgrass, for example, has two major ecotypes, upland and lowland. And there's also, of course, some overlap. There's also a coastal ecotype. So this is just a simplified model. And we chose one species from each of those ecotypes and stopped watering them for 4 weeks. And we found that the upland ecotype, which is called cave and rock, was clearly dying after a month without water. So on the left, you see the non-watered plant versus the well-watered plant, whereas the lowland ecotype was still very happy after the 4 weeks. So we sampled leaves and roots and ran different experiments on the samples. For example, we did transcriptomics via RNA sequencing. And first, we focused on the genes that had the highest fold change or differential expression under drought stress compared to the well-watered plants. So here you can see volcano plots of the differentially expressed genes after 4 weeks of drought stress in Alamo and Cave and Rock, and then it's the upper panel is leaves and the lower panel are the roots. And the yellow dot means that those genes were upregulated, the blue dots means that they were downregulated, and the black dots means that they didn't pass our significance threshold, so we assumed that they weren't as relevant for the drought stress response. And just by simply counting the dots, you can already see or estimate that there were less differentially expressed genes in Alamo, which was the more resistant lowland variety, than in Cave and Rock. And if you compare leaves and roots, that in the roots there were also more differentially expressed genes. So which might hint to that roots seem to be more responsive to the drought stress. And many expected genes we found, for example, lipid transfer proteins in the leaves, or AWPM19-like proteins, which are ABA influx transporters, and ABA is one of the big drought stress mediators, so that's not surprising. We're currently following up on some of those genes. We didn't see any terpene synthase genes among those very top candidates, so we decided to change directions a little bit and look into how specialized metabolic pathways are expressed under drought. So the terpenoid precursors are coming either from the mevalonate or MEP pathways, from the isopentenyl diphosphate via isoprene, the isoprene building blocks that I mentioned, form farnesyl pyrophosphate, which are the C15 molecules that go towards the sesquiterpenoids and triterpenoids, or via geranyl pyrophosphate or GGPP towards mono and diterpenoids. And this example, I just want to focus on di and triterpenoids, because those have been assumed to act in drought stress response, or have been shown to be involved in drought stress response in other plants before. Luckily, those key enzymes are highly conserved, so we could blast the switchgrass genome against our in-house databases for those genes and pull out all candidate genes for those key enzymes, and then compare their expression in Alamo, which you see in the left panel, to Cave and Rock. And here those numbers indicate the different sample groups, which isn't that important. what is important, or what was really striking us, is how similar the expression profiles are between Alamo and Cave Rock of those, let's call them for now, upstream enzymes. And also there didn't seem to be much of a drought inducibility. Did the same for the flavonoids, which derive from the phenylpropanoid pathway, and same idea here. Those are also fairly conserved. So we did a blast search of the switchgrass genome, pulled out the candidate genes, and again, there didn't seem to be much of a difference between Alamo and Cave Rock, and not much of a drought inducibility. So we walked a little bit further downstream into where the terpenoid networks are really branching out. And some of those genes we already had characterized. Some of the P450s and reductases we pulled out via genome mining again. And here you see a hierarchically clustered heat map, which organizes genes next to each other. Here you see all the gene candidates from this network here, organizes them next to each other if they are expressed under the same conditions. And D means that there was a drought treated plant, and C means control, and the number indicates the duration of the treatment. And then there's a separation into leaves and roots. And then you look for clusters of genes that are highly co-expressed. And you can already see that some of the genes are all only expressed in leaves, whereas others are only expressed in roots. So there's a clear separation here. And for example, here you can see under 2 weeks of drought stress in the roots, there pops up a really nice clusters of genes that were upregulated. And some of them have already been shown to act together in a pathway, and others then gives us the hint what will act on our already discovered molecules. And this is the Alamo, the lowland variety, which was very drought resistant. In Cave Rock, the cluster search was not as straightforward, which is maybe, this looks like a cluster here, but if you look closer, you can see that those are control samples from different time points. So that might potentially indicate that diterpenoids are not drought inducible in Cave Rock. Triterpenoid metabolic networks are typically more complex and difficult to construct, also less characterized in switchgrass. But luckily, most gene families are also highly conserved. So we did a genome-wide search again for the potentially involved enzymes, threw them together into a heat map, and I want to start with Alamo again. And what you beautifully get here for Alamo is this strong cluster expressed in the roots after 4 weeks of drought stress, which includes here color-coded genes from all steps of the pathway. Or for example, this smaller one in the leaves after 4 weeks of drought stress. And this is very helpful if someone now decides they want to go after switchgrass terpenoids, because this will be a great starting point for deciding which enzymes, for example, to co-express and to find the pathways. In Cave Rock, again, no great clustering was visible. And this got us thinking if maybe the diterpenoids and triterpenoids are in some way aiding Alamo to be so drought resistant. And to follow that idea, we looked at how the drought stress affect the metabolome. So in addition to the transcriptomics, we did untarget metabolomics via LCMS, which was done at Rob Last's lab at Michigan. And here you see a principal component analysis, or PCA, of all detected features, which passed our thresholds for abundance, which sums up to 4,000 different metabolites in leaves and roots combined. And so here you see the leaves before the treatments and the roots before the treatments. And the LMO control and drought stress plants cluster together, and the Kelvin rock control and drought stress plants cluster together. And same for the roots. So there's already a difference in the metabolome between those two varieties. And then after the treatment, you can see that the metabolomes shift apart. They still somewhat cluster in the ecotypes, but the treatment already has an effect on the metabolomes, which is a very common phenomenon. So what mainly contributes to the metabolome is the tissue type. If we would put leaves and roots together, you would only see differences between leaves and roots and not between treatments and ecotypes anymore. And then usually the ecotypes have a bigger impact than the treatment then. So we zoomed in a little bit further to identify the metabolites that are contributing most to those shifts. And here you can see a scree plot of the most important features which had the highest impact on metabolites under drought stress. In the leaves, you can see a lot of phospholipids. We couldn't annotate them further, unfortunately, but I think when it comes to untarget metabolomics, the annotations are always the hardest part. So phospholipids, we then assumed in the leaves are probably helping with the membrane integrity. Also, you can see a polyketide, which might be part of the leaf waxes, but I'm not an expert in this, and this is something Luke can better explain because he's the expert in cuticular waxes, so he might be able to tell us a little bit more. In the roots, you good. In the roots, you actually see one triterpenoid here being at the top and some diterpenoids that were increased under drought, especially in LMO. So when you look at the dots, it means like this diterpenoid, for example, went from not so abundant to very highly abundant, and from not at all abundant to medium abundant under drought. So instead of just looking at the top features, we wanted to see also what happened to all the specialized metabolites we were able to annotate. And here you can see another heat map. I'm sorry for all the heat maps you must look at today, which includes all terpenoids, flavonoids. We were able to annotate. So we were able to identify a good number of features based on their fragmentation patterns and mass ions on the LCMS, and by comparing them to our standards. We also included some metabolites that are known to accumulate under drought stress, like ABA or sucrose as our controls. And you can see that again, the metabolites clusters by leaves and roots, and this is what I already mentioned, that the tissue type has the biggest impact on the metabolite profile usually. And then they clustered by ecotypes, which was also not surprising. So it's LMO, control drought, Kelvin rock control drought, and then in the roots, just switched positions for the two ecotypes. And you get a very interesting accumulation patterns here. For example, a lot of flavonoids were only present in the leaves, or triterpenoids and steroidal saponins here were often either just in one of the tissues or one of the ecotypes. And for that, I would like to recommend this very interesting preprint from Jingjing Li in Rob Blast's lab about the fascinating diversity of saponins in different switchgrass genotypes. They tested more than just the two and found that there are big differences between the ecotypes. And they have also contributed the great metabolomics work for this project. But what you also can see is that those saponins or flavonoids weren't drought inducible. They were somewhat the same between the control and drought groups. So because compounds that are drought inducible are accumulating in alternating columns, like here the ABA or the sucrose, and also those 4 diterpenoid compounds that you can see down here, where you can see that they're accumulating highly in the roots of alamo and also of Caven rock, although a little bit less. And they didn't match any of our known diterpenoid compounds so far. So we decided to extract them from a ton of plant tissue, purify them, and expose them to NMR to solve the structures of the compounds. And this was another collaboration with the Tentillo lab at UC Davis and a former grad student from that lab, Amy Merrill. And so those are, I color-coded them. I hope you can see that. So you can see that those are actually diterpenoids, which you can never be sure of until you actually do NMR. So we were able to solve the structures here. And what we also saw that those molecules were the perfect fit with our already discovered diterpenoid network. This was the network I've shown you before. And 2 of the compounds nicely extend this branch here, coming from those Furano diterpenoids. And then one has a keto group added at the C2 position, and another one had another keto group added at the C14 position. And the other 2 molecules were stereoisomers of these 2 molecules. But we assume that they probably descend from a so far unknown precursor because it hasn't been shown that P450s are able to change the stereo chemistry of molecules. And because those names are pretty hard to remember, we gave them easier names. We called them panicoloids A to D. So now we're actually coming from the other side, from the end products. And now we're trying to find the responsible missing genes, the linking genes here, here, and here. But since P450s, the P450 families are often very conserved in their functions, we already have some promising candidate genes that we are now testing and hope that they will fill those missing gaps here. We are not the first ones who have found those compounds. At least 2 of them have been described before. You see Solidago arguda, which is commonly called the Atlantic goldenrod, or Croton campestris, which is used by traditional communities in Brazil for the treatment of health problems, or Veliciosa bicolor, which is also native to Brazil. And they have also found what we then named panicoloid A and C. Those papers are all a little bit older, from the last century, so they weren't wondering too much about the functions of those molecules, so this is still unclear. But similar compounds have been shown to have antioxidant functions. To figure out the functions of those molecules, we are now want to use antimicrobial studies and are using genetics, specifically VIGS. And VIGS is short for virus-induced gene silencing. So the basic idea is that you put your gene of insert into a plant viral vector, and you have to go a little detour through tobacco where this viral vector gets amplified. You do some tests, and then you can rub this tobacco sap, you grind it up and rub this sap into your little switchgrass plants, do some more tests, and then you want to measure the effect. And the idea is that the plants recognize the viral vector, the viral RNA, and through post-transcriptional gene silencing then degrades the RNA. and therefore also degrades its own RNA of the corresponding genes that you put into your construct. And if you do that, for example, with genes that are involved in chlorophyll or carotenoid biosynthesis, and those genes get silenced in the plants, you get a bleaching effect. So you already have a visible marker if you use genes, genes that will have a visible phenotype. So here you can see the wild type plant usually have these colors of the leaves. If you just inoculate the plants with an empty viral vector, you already get a mild bleaching effect, which is due to the impact of the virus on the plants. But for some of the genes, you can see a pretty strong bleaching effect. And if you look at the gene silencing effect, you can see that in the leaves as well in the roots, we had a silencing effect between 60 and 90%. So those are pretty exciting recent results because gene editing techniques in switchgrass are still somewhat rare and complicated. So now after we identified key genes and molecules for stress tolerance in switchgrass, we can now, in the next step, use, for example, VIX to confirm the gene functions. And as a next step, or hopefully somewhere in the future, we can then use precise genetic engineering to create a desired blend of specialized metabolites towards more resistant crops. And with that, I would like to end with a huge thank you to all the fantastic people I had the honor to work with over the past 2.5 years at UC Davis, especially my PI, Philip Zerby, and the other members of Team Switchgrass. I would also like to thank our collaborators at UC Davis, from the last lab at Michigan State University at the Berkeley Lab, our sponsors, the German Research Foundation, the US Department of Energy, and the Joint Genome Institute. And I would like to end with some shameless advertising I think I unfortunately stopped sharing my screen this second. So, or can you still see my screen? I don't know what happened. I'm sorry.
You can only see your background.
So I'm okay. I see.
You're still sharing your screen.
Yeah. Sorry. That's because I wanted to do shameless advertising.
Yeah.
Because I'm starting my own group at the University of Groningen in the Netherlands next month. And if you are interested in working with me, no matter if you're a student or interested in collaboration and would like to combine genomics, metabolomics, genetics, but I would also like to bring in more and more the physiological role of those molecules to better understand stress tolerance. Please feel free to reach out. Or if you just want to maybe find your own group of molecules and get to name them, which is also fun. Yeah, please feel free to reach out and stay tuned. And thank you very much for your attention.
Yeah, thank you very much, Kira. Great talk. Thank you. And I just wanted to highlight again that we are recording this session. So if you don't want to be in this recording, please keep your camera off. And we are welcoming questions. You can post the questions in the chat, or you can just like raise your hand, and then we will go through the questions.
I had a question about, I probably missed this part. You mentioned that the roots were showing a lot less differential expression compared to the leaves in under the stresses in both the cultivars. I was wondering if you saw a similar pattern for metabolomics.
Sorry, I don't know if I said it the wrong way around. So what I was trying to say that there are more differentially expressed genes in the roots than in the leaves.
Oh, okay.
I don't know if I can just jump back a ton of slides to this very first volcano diagram where you can see that in, let me get the laser pointer here, for example, in the roots, there are more differentially expressed genes than in the leaves in the upper panel. Maybe it's a little bit hard to tell by the dots, but yeah.
Do you see a similar pattern for the metabolites too?
Exactly. Yeah, there were definitely more changes in the roots for the metabolome. And there could be multiple reasons for that, right? So it could be that the stress response just simply starts in the roots. Oftentimes we don't even look at the roots, but we see a change happening in the leaves. But of course, in the roots, there's a lot going on trying to get more water in by improving root hair, root growth, all of these things, maybe attracting the right microbes that can help you to increase your uptake of water. So I think in many ways it makes sense that the plants in times of drought stress really focus on their roots.
Yes, I just mentioned that because we are seeing the same thing in Brachypodium.
Oh, great. Great. Have you looked for diterpenoids?
Not yet.
If you need any standards or so, let me know. It would be great to know if diterpenoids seem to be important in other crops too.
Hi, Kira. Thanks for the great talk. I have a question, a more technical question about your VIGS technique. I've never come across the rubbing technique. Can you expand a bit on that, using benchimano first and then applying it to your leaves? It looks like an interesting application.
Sure, yeah. The problem is that other than tobacco, which has this very soft, spongy leaves almost, where you can just infiltrate them with Agrobacterium, switchgrass has very sturdy, hard leaves. So you wouldn't even get the agro-infiltration into the leaves. So you really have to increase the viral titer in the tobacco plants. And then we grind up the plants and add something that's called mesh carborundum, which is basically what you have on your sandpaper, but without the paper. And then we use a cotton swab or something to dip that into that mixture, this viral mixture, and really rub it into the leaves. You kind of have to damage the surface of the leaves to get the virus in. But once it's in the leaves, it spreads systemically. So we actually don't test the leaves that we rub inoculated, but leaves that are newer, for example, or the roots. And we weren't the ones that invented it. I think it has been first tried in corn, if I don't misremember. So I think this is a technique that has been used in other monocot crops too.
Thanks. And I also have a second question. I noticed that your DXS and SQS genes were highly upregulated. Do you think that there's a flux control in response to the drought there?
Yeah, that is a good question. So far, just looking at the gene expression, you would almost think that there's somewhat of a constant flux going towards the more downstream products, which didn't seem to be too regulated to us.
Hi. So I think you mentioned some of the potential ecological role for these molecules. I wonder, have you do any bioactivity assay to test their function for the diterpenoid you discovered?
Yeah, that's a good question. And that's always, of course, almost the most interesting question, right? So how do they work? The problem is that for example, to do bioactivity assays, you need a lot of the compounds. And as you can imagine, to get them in a pure enough, you need to extract from a lot of plant material. So I think once we have the full pathway towards the molecules and can simply express them, it will be a lot easier to do bioactivity assays where we can produce bigger amounts of the compounds. So right now, to answer your question, we haven't done any bioactivity assays, and those are also somewhat recent results. But yeah, I would love to do that, and I could imagine, if I would just speculate, that those compounds either have antioxidant functions, as I mentioned, for similar compounds, or that they maybe interact with the microbiome. And for that, we could, for example, test if they're being exuded, right? That would be the logical next step. And then test them against some known microbes from the switchgrass microbiome. Yeah, good question.
All right. Just gonna check the chat real quick if there's more questions there. Does not look like it. Are there any more questions from the audience? We have enough time for.
I have a
Oh.
Sorry, yeah, that was a great talk here. Really enjoyed it. I know a little bit of the origins of this project with switchgrass. So I'm curious. It's a very general question. So with your new faculty position, your new research group in Groningen, are you continuing to work primarily on switchgrass, or are you moving into other systems?
Even though I would love to keep working with switchgrass because it has turned out to be so interesting in terms of terpenoid metabolism, the Netherlands are not too interested in switchgrass as a bioenergy crop, unfortunately, so it would be hard to get funding there. So I will continue working on switchgrass as a collaboration or in collaborative projects, for example, with UC Davis. In the Netherlands, I'll be more focusing on crops that are relevant there. For example, terpenoid metabolism in potatoes, tomatoes, and fescue grass, which is in some ways very similar to switchgrass, right? And it's being used, for example, as grass on golf courses, football courses, but also in the dunes there next to the coastline. So.
Great. Fantastic. Hey, good luck with your new faculty position. Yeah, I'm sure you'll be doing really well.
Thank you very much.
Okay, sure, thanks.
Yeah, Elizabeth, please go ahead. Your question.
Yes. Can you hear me?
Yes.
Okay, cool. Just the video. Hi. Yeah, it was a very interesting talk. Thank you. And in these experiments where you compared drought versus control in your metabolome analysis, did you also possibly see some polyamines coming up? Polyamines?
So this was not really our focus, unfortunately. So because it was so hard to annotate all those thousands of compounds, at some point we had to focus on what we had standards for and what we could annotate with our databases. So unfortunately, we didn't look.
Okay, thank you.
Thank you.
All right. Thank you everyone for all the questions, and thank you, Kira, for your great talk. If anyone wants to stay on for a few more minutes for more informal chat, please feel free to do so. And otherwise, I would like to thank everyone and see you next time. Oh, I said Sandra. Different account.
Great talk, Kira. Welcome to the Netherlands.
Thank you. Are you the postdoc that Lars mentioned who just recently moved to the Netherlands?
Most likely, unless he knows another one. Yeah, you're in Groningen, right? Or like
Yes.
you will be. Yeah, okay.
Yeah. And I can see you already know how to pronounce that, so you have been practicing some Dutch.
Yeah, two of my really good friends who are in chemistry, they moved to Groningen about 1.5 years ago. So they very much enjoy it.
Okay, great to hear.
Yeah.
Yeah, so far I've only been to the Netherlands for vacation, so I'm not sure yet what to expect for